It is confusing. And remember that in the spinal cord, the gray matter is deeper and the white matter is more peripheral. It's just the opposite in the brain.
The very top right-hand corner, it changes back to gray matter, correct?
Yeah, the very right, absolutely. And so right here is granular. I can't spell it all out, but that's granular. White or the gray matter.
Is there a definitive demarcation between the white matter and gray matter, or is it just kind of like a slow progression of the white matter?
Yeah, it's a slow, you know, in the cerebellum it looks very unique and very definitive, but it's probably a slow progression. Okay. If you think about it, what is white matter? Myelinated axons, okay? And so if you think about it, okay, you've got these cells in the granular layer, which are probably interneurons. Did Dr. Defoe, I couldn't be here for that lecture. Did he talk about interneurons? Okay, so those, a lot of these cells in this granular layer could be interneurons, you know, and they're going to send their axons out into white matter. Right. This is just, again, a silver stain, just showing the Purkinje cells, and you get a sense of the dendrites coming from it. I'll back up a second, too. The Purkinje cells might also be sending axons out into the white matter. They could be coming all the way through the granular layer and sending axons out into the white matter. This one is not such a big deal. It's just to highlight these cells, the pyramidal cells. Okay, and these are the dendrites on the pyramidal cells. That's not something that's that important. And all these astrocytes, we're not going to ask you to identify the astrocytes. You know that you would have fibrous astrocytes versus protoplasmic astrocytes. Protoplasmic astrocytes are associated with gray matter. Fibrous astrocytes are associated with white matter. Yeah, PG is the way I remember it. Because, you know, I'm not a neuro person, and that's just a simple way to remember it. Okay? These particular cells, and again, I'm not going to ask you to be able to identify them. These are probably oligodendrocytes, which in the central nervous system, they play the same role as Schwann cells. They invest the axons with myelin. And one oligodendrocyte is thought to invest multiple axons, which is why it looks like it has these little things coming off and projecting, maybe almost imagining that they're wrapping around some of the other neurons. And again, you'd never be able to recognize this just that way, but these cells are microglial cells, which are macrophages in the central nervous system. I think that these are melanin granules in neurons, probably in the locus ceruleus. Melanin granules are a biosynthetic product involved in, this tyrosine is involved in that, and that's how we get some of the synthesis of dopamine. And when you're looking around on a glass slide, you can find these very characteristically in certain regions of the brain, like in the palms. Sometimes you'll see in the lab write-ups, look for residual bodies or lipofuction granules in neurons. Those are very difficult to see, and I would never ask you to ID them. Even on the University of Iowa site, they label them and you still can't see them. Okay, so don't worry about trying to identify lipofuction granules in any of the tissues. Just understand what they are. They're just end products of lysosomal digestion. These are some of the touch receptors near the surface of the skin. This is all a stratified squamous. Little layer of keratin above it. This is the dermis. These are dermal papillae, where these receptors reside. If you think about the dermis and epidermis, they interdigitate with each other. So we could call this region of the epidermis, we call it an epidermal peg. I don't care that you remember that, but that's interdigitating with the dermal papilla, the underlying connective tissue. It's just a way to help to anchor, increase surface area of the epithelial sheet, to anchor the epithelium to the underlying connective tissue. And that's just a higher power view of these Meissner's corpuscles, as opposed to that one, which is a Pacinian corpuscle. Okay, and the very tiny pink spot in the middle is actually the axon. Okay, both in this one and in these, as you get near the very end of the neurons lose the myelin sheaths. Okay, these are touch receptor, so very fine touch on the fingertips. These are deeper, kind of, where the dermis and the hypodermis come together, and those are more like pressure sensors. And I know this is hypodermis because I see all the white cap.
The circular rings around all those cells right there, are those supporting cells? What are those?
They're fibroblasts, probably, and they're just making these concentric rings, and these concentric rings of tissue in real life are just filled with fluid. Okay, so it's like a mechanoreceptor. Okay, that when you're pressing, it's firing, and it's just sensing pressure.